Okay, chapter 3. Chapter 3, remember from the beginning, I told you that we're going to start from the smallest level and we will building up, right? Starting with the atom and molecule and macromolecule, right? And then we go organelles, cells, and when you put the cells together, you make tissues. Remember this? System, and then the whole body, right? This is the sequence that we're moving. So our next step here is the cell. Um, so it's about the cells, and these are the outcomes. You can read it. Basically, we're learning about the cells and all the structures of the cells. Cell theory, this is where they talked about the cells, and cytology is two parts. Cyto is a prefix that means cell. Ology means studying. Cytology, studying cells. Okay? And basically, we have two different types of cells. Number one, the normal body cells. And body in Greek is soma. Soma, translation. Soma is body. So somatic cells, the body cells. What do you mean the body cells? All cells in your body minus sex cells. Okay? So sex cells is alone. It's not, we will talk about it later. But for now, we're going to start with the somatic cells, which is every single cell in your body that's not the sex cells. What is the cells? Cells consist of this membrane outside the wall. It's not called the wall, it's called the membrane that's covering the cells. So this is like we're in this room here. You have all these walls. This is a membrane. And we have students and me and chairs and desks, right? You have a lot of things. Where are we located? All of us here, where are we? We are inside the cell in this room, right? The inside is called cytoplasm. Okay? So if this room was empty, there is absolutely nothing. This is an empty room, right? So this is the cytoplasm. Outside is a membrane. Inside is the cytoplasm. Is this cytoplasm empty? No, it's not empty. Okay, it contains watery fluid. I didn't say water, watery. It's water and other things. What do you call it? Cytoplasm, the plasm of the cells. What do we have in this cytoplasm? We have different organelles. Do you mean organ? No, organ is like kidney and heart. This is organ. Tiny organ is organelle. Do you mean E-L-L-E means tiny? Yes. Heart is an organ. Mitochondria is organelle. You're adding E-L-L-E at the end, which means the tiny organ. So we have a lot of these organelles inside. Something like the nucleus is the largest organelle. We have Golgi apparatus, we have lysosomes, mitochondria. We will talk about all of those. These are the different things. We will go them one by one. Okay, just to know that these organs that we're going to talk about, some of them are surrounded by a membrane because the contents are either important or dangerous. In both cases, you have to surround it with a membrane to keep it and to save it. Others are not dangerous. Like if I ask you, where do you keep the oven cleaner? Do you put it like in a cup anywhere? Of course not, right? Anybody can, it's dangerous, contain dangers, right? So you have to put it in a bottle and you have to like close it. So you put it inside a membrane, right? Versus anything else, it's okay. You can put it like in any sugar or something. It's not something that's harmful or something, so that's okay. In this point specifically, you will see questions. Which of the following is membranous? Which of the following is non-membranous? So you need to know them by name. Which is which? So we have all of these. We will have the cell membrane outside. The cell membrane, and then if you go inside, you will see different things like peroxisomes, lysosomes. You will see nucleus and other things. So, but let's go one by one. Here is the nucleus, and let's start with the plasma membrane. Plasma membrane. What's a plasma? Plasma, the inside. The inside is watery. Do you agree on that? It's watery. And the outside is also watery. Here is one cell. Okay? What do we have inside? Watery cytoplasm. Here is another cell. What do we have inside? Watery cytoplasm. How about the outside, in between them? Watery fluid as well. So there's water inside and water outside. For that reason, if you remember the phospholipids and glycolipids, we talked about those, you will see how they are going to face this watery medium. So this is plasma membrane that's separating and surrounding the cells with all the components from the outside and separating the inside from the outside. So why do we have the membrane? It's isolation, physical isolation, just like this wall. The room is isolated, acting as a barrier. And because of that, it regulates what goes in and what goes out, right? If you don't have this wall, anybody can go in, anybody can go out, right? But since you have a wall and you have a door, certain people are allowed in here and others are not, right? So regulating the exchange, what can go in, what can go out, and what cannot, all right? Um, it will also give you an idea about what's happening. Like if I put cameras out there, and I can see like people walking and somebody jumping in there, and right? If you put cameras outside, there is something that's kind of like a camera outside the cells that telling the cells what is happening outside. We will see it. The last function is the membrane. There are things that are holded to the membrane. So it is structural support. This is generally speaking. What is the main bulk of the membrane? What is the main bulk of this wall? Bricks, isn't it? Bricks, and you put some cement in between. But what's look at it here. What's the main thing here? Isn't it bricks? Right? For the cells, it's phospholipids that we talked about. This is like the bricks. Is that the only thing? No, this is the bulk. The big bulk, 90% of the membrane is phospholipids. And phospholipids, we talked about it, if you still remember. The head, the one that contains phosphate, is hydrophilic or phobic? Hydrophilic. And the tail, hydrophobic, because it's fatty acids. And we put it like this for a reason. I'll talk about it. Is that it? No, there are other things, including proteins, called membrane proteins. Some of them are called integral. What do you mean integral? Integral means integrated inside, integrated, and also for integrity of the membrane. So integral, integrated inside, and then peripheral, that's outside. Clear? This is a membrane. If I have protein in here, it's integral. It's integrated inside. If it is bulging out like this, coming out of the wall, inside or outside, we call it peripheral proteins. Why do we have proteins? Why don't we just have phospholipids, which is the main bulk? No, you have to have proteins for different reasons. Some of them are going to stabilize, act as stabilizers. Okay. Some of them will be as identifiers. What's identifiers? Like if you come to this room, doesn't it say the room number? So you know where you're going, right? If you don't have numbers, you wouldn't know, right? This number outside, compared to the cells, protein outside of the cells, to tell everybody else, who are you? Are we following? It's called identifiers. So there is a protein outside on the surface that's telling everybody else, who are you? Are you a liver cell? Are you a skin cell? Who are you? Are you a blood cell? Okay. So like everything else around the cells, it can't like see like us. Like look at this. Obviously, this is red blood cells. Yes, you see it from outside, but inside they don't know. The only way for them to know is look at this identifier. And they will also, proteins, beside those two functions, they also work as enzymes. So there are some reactions happen around the membrane, so it will work as enzymes. Some of them will act as a receptor. What do you mean receptor? I mean like the door ring. When somebody come to you, they push on a ring, right? And you know somebody wanted to enter, right? This ring is protein, and we call it receptors. So something is coming to you, it will go to the receptor outside, and you're going to receive it. Yes, why are you here? I have a delivery for you, for example. Okay, I will take it. Or I have something that I'm gonna put it inside your house. Okay, go in, right? So these are the receptors proteins. Carrier proteins in the wall, there are some proteins act as a carrier. Like I want to get this protein inside, another protein or something else. I want to move it inside. Okay, move. No, I can't move. It's too heavy. Okay, there are some proteins that will take it from outside and carry it inside. These are called the carrier proteins. Another thing is some of them have a channel like this. Not this is a protein, okay? This is another type of protein. It have channels. So like water, how can the water like go in? The cells need water all the time. How does it go? There are channels for them to go. Otherwise, like how can we go inside from the door? There is a door, right? But can we pass from the wall? Of course not, right? For the cell membrane, these bulky phospholipids and proteins, they do not allow anything to go through. But there are some channels for certain things to go. Are you water? Yes, go through your channel. Phosphate, sodium, there are certain things that are needed. You have a channel for it, channel within the protein. These channels, this is a channel, right? Some of them have a gate like this, open, close, like this door, okay? And others, like in the back here, if you don't have a door at all, it's just an opening, right? This is called non-gated. So some of these channels have gates and others do not have gates. So this is for the proteins. So what did we say so far? What's the main bulk? Two layers of phospholipids. Two layers of phospholipids. And we know what phospholipids. This is like the bricks. Is that it? No, we have proteins. We talked about it. The next is carbohydrates. We do have carbohydrates, but it is mixed with one of those two that we talked about. We talked about proteins and lipids. Are we following? We talked about proteins and lipids. How about the carbohydrate? Yes, carbohydrate is not carbohydrate. It's not pure carbohydrate. No, it's mixed. Like glycoproteins, proteoglycans. Remember this? Can you tell me which one of those, like proteoglycans, which one is more? Protein or sugar? Sugar. Very good. Yes. And glycoprotein is the opposite. And also some glycolipid. What's glycolipid? Sugar and lipids, right? So why do we have these carbohydrates out there? These carbohydrates outside, they call it glycocalyx, and you need to know that. What's glycocalyx? Glyco is sugar, right? We agreed on that. Glyco. Calyx means sticky. Sticky. Calyx means sticky. So what's glycocalyx? It's a sticky sugar. Why do you need something sticky outside? Because certain things, when they come to the membrane and they want it to work on the membrane, you want them to stick until they finish the job and then leave. That's why you have it sticky. Okay? What is this sticky coat? Glycocalyx. Glycocalyx. Why do we have it? Because you want things to stick in there. And also it works as lubricant. If the cells come together like this, and this is sticky membrane, but it is soft, and this is sticky membrane, they are not going to come in contact with each other. They're not going to touch. Okay? If something come and stick itself, that's fine. But otherwise, it's lubrication. When they come close to each other, this is lubricating so they do not have friction with each other. Anchoring is what I talked about. Anchoring. What do you mean anchoring? Like something goes stick itself. So anchoring. Um, Specificity, they also help receptors. Remember I told you there are some proteins that are receptors, work as receptors, like the ring on a door. Some of them contain also carbohydrates. Recognition for immune response, same thing. What is it? Let me explain it a little bit. Your cells, every cell in your body, they have an identifier outside, right? So that everything else will know who you are. If you have a mail delivery, how they can know, they know that it's you that I should deliver, because there is a number on your house, right? 34555 Ford Street, for example. This address is our recognition, or this receptor outside is for recognition. Part of it is glucose. I mean carbohydrate, I'm sorry. But how does it help immune response? I'm not going to go into the details, but let me tell you this. Inside your blood, you know the white blood cells, of course, so, right? White blood cells for defense. Defense like what? Like they attack any foreign organ or foreign cell, any foreign substance, anything that's not belong to your body, right? Anything that's foreign, we should attack it. What are you doing here, right? So how do they know? In the beginning, when we are embryo and fetus, the immune system got a memory like this. You're the immune system, right? Yes. You're in charge of defending us. Yes. Do not attack any of the cells. How to know? We will put an identifier outside, and I want you to memorize it. When you see this identifier, this is your red blood cells. Don't attack it. When you see this identifier, it's your liver cells. Don't attack it. So this is the recognition that I'm talking about. So if you got like different red blood cells donated, that's not your type, you know what's going to happen? You're going to attack it, right? Anything that's foreign goes to inside your body, the immune system will say, what is this? Let me look at the identifier or the recognition. Is it liver cells? No. Blood cells? No. Skin cells? Oh, you're foreign. Attack. Kill it. Okay, it's programmed like this. So I mentioned in the beginning that the main bulk of the membrane is a double layer of phospholipid, right? Can you see this? Look at this orange. This is the phosphate. The head, is the head hydrophilic or hydrophobic? Hydrophilic. And here is the tail is inside, which is hydrophobic. But you have another set or another layer. Look at the head down. And what's inside, deep inside, it's the tail. Did you get the idea? Head, tail, head, tail. You put them like this. So where are the heads? Facing outside of the cells, which is watery, facing the inside of the cells. Where are the tails? The tails are facing each other. Is that clear? This is how we arrange it for a reason. This layer outside here, you are interacting with this watery medium outside, right? The watery medium outside. In order to interact, you have to be hydrophilic. How about this one here? Why did you put it like this? Like upside down like this. Why do you have the heads down here? Because the cytoplasm is watery, isn't it? Right? If we are right here, okay, all of us are working here. And there are like Japanese people out here and French people out here. Who of us can understand Japanese? Me, you will be here to deal with it. Okay, and on the other side, same thing. Inside, it's okay. But in order to interact with the outside, you have to have something that understand their language. It's water. So you have to have hydrophilic, right? And that's why you put it in this way. So this is the main bulk, the orange and the blue. You will see some of these integral proteins. Look at these integral. Integral means within. Look at these, within. And you also have these peripheral. What's peripheral? Like outside. Peripheral is outside, not inside, not within the membrane, outside, peripheral. So these are the different types of proteins. Some of them will have a channel like this. Look at the channel. This is gated channels. Look how it's closed. You see the channel? If something come here, you have to open the door for it. These are called gated channels. How about the cytoplasm? Cytoplasm is two parts. Right now, if I want you to describe everything in this room, in this classroom, you will say this is an empty space that contain students and chairs and desks, right? The empty room is called cytosol. It's not empty. It's just contain watery fluid. Clear? Without anything, just like this. Cell, the inside contain watery fluid. That's it. I didn't talk about anything else. No students in here. Nothing else. Just inside of the cell that contain watery fluid. That's it. What do you call this? Cytosol. Cytosol. What if you add the different organelles, like with the students now? Cytoplasm. Did you get the difference? Cytosol is just the watery medium inside the cells. How about the cytoplasm? Watery medium plus everything else. And we will discuss what I mean by everything else. Now I mentioned, what's a cytosol again? The watery fluid inside, right? Watery fluid. Is it water? No, it's watery. It's watery. So what do you mean by water? It's water and other things. What are the other things? The other things are proteins, ions, nutrients, waste products. All of this is dissolved in the water. Okay? How about the outside? Isn't it watery as well? Outside is watery and almost the same like this. What's the difference that you have to know? Inside is watery fluid, outside is watery fluid. Is it exactly the same? Very close, but not exactly the same. And you do need to know the difference. Okay? Watery fluid, inside, outside. What's the difference? The difference is the inside contain more potassium than the outside, and the outside contain more sodium than the inside. Do you mean the inside contain potassium only? No, I didn't say that. Do you mean that there is no potassium outside? I didn't say that. You do have potassium and sodium inside. You do have potassium and sodium outside. Clear? Both of them have the same thing exactly. It's all the same, but different amounts. Potassium inside and outside. Which one is more? Inside. You have to know this. Potassium and sodium outside. Which one is more? Sodium. Is more outside than inside. Okay? Proteins also are more inside the cells. So what's more inside than outside? Potassium and proteins. Okay? And then with the cytoplasm, with the cytosol, if you add the organelles, that will become cytoplasms. Organelles. Organelles, I told you E-L-L-E means tiny, right? Organelles. Some of these are membranous and some of these are non-membranous. Definitely a question or two. At least is a question. How the question would be, which of the following is membranous? Which of the following is non-membranous? Or all of the following are membranous except. So you need to read the question to understand exactly what I'm asking you. Membranous. Which ones? Endoplasmic reticulum is membranous. Golgi apparatus, lysosomes, peroxisomes, and mitochondria. Okay? How about the non-membranous? Cytoskeleton is non-membranous. It's not surrounded by membrane. Centrioles, ribosomes, proteasomes, microvilli, cilia, and flagella. Okay? Thank you. Okay, so you do need to know this. I can give you a question like this. All of the following is membranous except endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, microvilli. It's microvilli. The only one that's non-membranous. Okay, this can be a question, but you have to know it. It's just an example. Which all of the following are membranous. All of the following organelles are membranous except. And then I gave you the answer: A, endoplasmic reticulum; B, Golgi apparatus; C, lysosomes; D, peroxisomes; E, microvilli. Your answer is E. Microvilli is non-membranous, isn't it? It's right there. Did you guys get the idea? Okay, so let's go through them one by one. The first one, which is non-membranous, is called cytoskeleton. Translation: cyto means cells. Skeleton, skeleton, just like the skeleton that we were doing today. Skeleton. The skeleton of the cells is 1 out of 3. Do you remember the skeleton that we did? There are tiny bones and big bones and small bones, right? Same thing here, but on a smaller level. So what do we have here? From small to large: microfilaments, intermediate filaments, microtubules. Obviously, microfilaments are the smallest. Intermediate, from the name, it's intermediate. And then the largest is microtubules. Microfilaments, these are the tiny ones. What kind of protein, or what's the structure? Actin. Do I need to know that? Yes, actin. It contains actin. And it's going to interact with another protein to make some sort of a movement that's called myosin, and we will talk about it. But this is the smallest ones. Okay, what does it contain? Actin. It's going to interact with what? Myosin. It contains actin to interact with myosin in a contraction. Next one is intermediate filaments, and the name gave it away. It's called intermediate. So what's the size? Intermediate, mid-size. So this is the mid-size one, and it is durable to strengthen and stabilize. Microtubules, these are the largest, and they contain a protein that's called tubulin. Okay? What kind of protein in microfilaments? Actin. What kind of protein here? Tubulin. And it's called micro what? Tubules. Tubules, tubulin. And these microtubules, we will use it later when we talk about something that you already did in general biology. You guys did general biology, right? I think it's 131. So, of course, you did this prophase, metaphase, anaphase. Remember the spindles that attach and contract to separate? This is microtubules. Here is a cytoskeleton. Look at this. It's different things like these. The purple one or the pink one are intermediate filaments. Look at the microtubules, the yellow, it's bigger, like this yellow right here. And look at the microfilaments, the smallest. And they extend into the finger-like projections coming out of the cells, not all cells. Some cells are just like this. Membrane, and that's it. Okay? Some of them, according to the function, and again, I'm not going deeper than that. Just understand this. Some of them are like this. Just a membrane, cytosol inside, organelles, you call it cytoplasm, just like this. Some cells, because of their function, they have finger-like projections like this, projecting out of the membrane. And these are called villi or microvilli. These microvilli contain microfilaments. Why do we have the microvilli? Because this is our next organelle. Microvilli is a finger-like extensions, right? Instead of being like this, you will have it like this. So the membrane will go like this. Compare this membrane, look at this. If you're measuring now, look, all of this versus this. Of course, this surface area is a lot bigger, isn't it? From here to here, let's say 2 inches, right? How about this? 1, 2, 3, 4, 5, 6, 7, 8 inches. Isn't it a lot bigger? So this is to increase the surface area. That's why we have microvilli. Centrioles is part of the uh, is part of the cytoskeleton, and it will make the spindle apparatus attaching to the centrosomes. It's part of the cell division that we will discuss later. Cilia and flagellum is also extensions. Okay, what's the difference? Like, how many types of extensions do we have? Here is the normal cell. Okay, no extensions. Some cells have extensions. The first type of extension is villi, finger-like projections to increase surface area. Clear? This is what we have to know about it. Because if this is the membrane, the surface area here is small, right? Surface area for what? Things going in and out. You don't need to interact that much. But if you do, you have to have extensions like these to increase your surface area. This is one type. The other type of extension is called cilia, and the cilia is basically to move things around, not to increase surface area, like the villi. What do you mean move things around like this? If this is the cell, and these are the cilia, when they move like this, they making more contact outside are going to make more contact just by moving them around. This is like if you have some water here, okay, and you want to drain it out. You make an opening here, right? It's going to drain, but can you push the water like this? It, if this is like in your backyard or something, you have water like this, and you wanna move it out. Okay, you can just push with your hand like this to help it, right? This is what's this doing. It's just moving the fluid around to have more interaction, and it can pass more. And these cilia are movable because they wanted to move things around your cells. Flagellum is the last type of extensions, and this is completely different. Flagellum is basically a tail. It's whip-like. Did you see the picture of a sperm, for example? There is a tail, and when this tail moves, it helps it to move. So this is usually one extension different than the villi and cilia. Are we following? Villi to increase surface area. Cilia to move the outside structures around. Flagellum is one extension that looks like a web shaped, like the tail of the sperm, to help the cells to move. This is how the microtubules looks like. And these are the cilia. And obviously they contain the microfilaments for movement. And look at this picture right here. It's on the surface like this. Next organelle are called ribosomes. Okay, we're talking about the non-membranous now. Or ribosomes. What are the ribosomes? Ribosomes are exactly like the machine inside your factory, right? If you wanted to make, if this is a factory making TVs, right? The machine that do the work for you. This is the ribosomes. For specifically in the cells, this is the machine that makes the protein. How about the DNA and messenger RNA? No, DNA is the manager. Doesn't do anything. It just manage. RNA workers, they help you. But where's the factory itself? The units, the machine. The machine is ribosomes. Okay? So what's the ribosomes? This is where we make proteins. This is the machine that help you to make the protein. Okay? And there are two types of these ribosomes. These machines are two types. One of them is scattered in the cytoplasm. We call it free ribosomes. And others are attached to another organelle that's called endoplasmic reticulum. What does it mean? Endo means inside. Are we following? Are you paying attention? Another organelle is called endoplasmic reticulum. What does it mean? Endo means inside. In. Plasmic, plasm. In the cytoplasm. What's reticulum? Translation. Network. Network. What's endoplasmic reticulum? Network in the cytoplasm. That's it. A network in the cytoplasm. So ribosomes, these machines can be free. Some of them are free, scattered, and others are stuck on the surface of the endoplasmic reticulum. Why do you have two types? Didn't you say that these are all machines to make proteins, right? Yeah, some of them are free, some of them are stuck. What's the difference? The free ones make protein for you, for the cell itself. The one that's stuck on the surface make protein for export. We know that all these, like for example, car factories, we know that they make some cars for the country, for America, and others for export, isn't it? Same thing here. You have some factories or some machines, in this case, that make protein for you. Which one? Free. They make protein for your own use. And the other ones stuck on the surface of the endoplasmic reticulum, they make protein for export. Why did you put it on stuck on the surface like this? Because when you do the protein, it will go in this network and go out for export. Did you get the idea? The difference? Are we following? Okay. So free ribosomes, these are free to make the protein for you, for the cell itself. The fixed one to make protein for export. You're going to package it, put it in a nice package, and export it out to go to other cells to use. And if this is endoplasmic reticulum, and we know what endoplasmic reticulum is, network inside the cytoplasm, isn't it? This network, if it doesn't have anything on the surface, we call it smooth endoplasmic reticulum. Are we following? Smooth, it doesn't have anything. Smooth. How about those that have ribosomes stuck on the surface? Is that smooth? If you have this stuck on the surface. No, it's not smooth. We call it rough. So what's the difference? Rough contain ribosomes stuck on its surface, and it looks rough. It's not smooth. Next one is called protease, proteosomes. Cut it into two pieces. Protease, somes. What's somes? Somes, soma. What does it mean? Body. What's protease? I told you the suffix. It has ase at the end. Enzyme that work on what? Protein, exactly. So protease, enzyme that work on protein inside the body. Proteosomes. Proteosomes. So why do you need enzymes that break down proteins? Why do you need it? Because don't you keep on recycling your structures inside your cells? You keep on, if this structure here is worn and wear and tear and it's not usable anymore, what are you going to do with it if it is protein in nature? Send it to the proteosomes, break it down, recycle the protein. You don't throw it away, right? You don't throw it away. Reuse it. How? Break it down into just proteins and use it. So who is going to do that for you? Proteosomes. Membranous. We have endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and mitochondria. Endoplasmic reticulum, I just talked about it. Reticulum means network. Endoplasmic reticulum is a network inside the plasma. What do you have it for? Well, let me say this. I can't ask you this question. I can't ask you, what's the function of endoplasmic reticulum? You will ask me, which type are you talking about? The smooth or the rough or both of them? Which one? Does it have a different function? Yes. It's like this. If it is rough, what does it have on the surface? Ribosomes. And what's the function of the ribosomes? To make proteins. So what's the function of the rough endoplasmic reticulum? Proteins. Make proteins. It make proteins. By itself? No, by the ribosomes on top of it. But it take it. Did you get the idea? Here is a network inside the cells. Because of the ribosomes stuck on its surface, what's the function? Making proteins. Itself? No, by the ribosomes. But the ribosomes is stuck on it. It's part of it, right? So it will make the proteins, and then the protein will go inside this network, moving, put it in a package for export. It's not going to be exported yet, not ready yet. But at least for now, you run within this network. Okay, ribosomes make the proteins for me. Ribosomes make it. Okay, give it to me. I'm going to take it and put it in a package and send it to the shipping unit. They will ship it out. It will be the Golgi apparatus, and we will talk about it. The other one that, so what you see here is both of them combined. But most of the time, I ask you separately. So you do have to understand which is which. Okay? So rough endoplasmic reticulum, protein. Smooth endoplasmic reticulum, it make carbohydrates, lipids, and detoxification. Carbohydrates, lipids, detoxification. Rough endoplasmic reticulum, proteins. You make proteins. Smooth endoplasmic reticulum, carbohydrates, lipids, detoxification. Is what I'm saying clear? Here is the smooth. What do you want us to know here? First of all, it's smooth. It means there are no ribosomes, right? Since there are no ribosomes, it looks smooth. And the function is not making proteins, right? So what's the function? Making lipids, carbohydrates, different types of lipids, different types of carbohydrates, and detoxification. How about the rough? I already told you. We make proteins, right? And we make proteins where? In the ribosome stuck on the surface, right? What do you do after you take proteins from the ribosomes? I'm gonna pass it. I'm gonna put it in a nice package, but this package is called transport vesicles. Where do you put the protein when you get it from the ribosomes? I put it in the form of package called transport vesicles. Okay, look at this. Do you see this size? This side? Can you guys see this side? This is smooth, right? And this side is rough. The rough here make protein and put it in the form of these vesicles. The smooth carbohydrates, lipids, um, and detoxification. Next organ is Golgi apparatus. What's the Golgi apparatus? Remember I told you that the endoplasmic reticulum, um, it makes proteins, right? Endoplasmic reticulum makes proteins, the rough, right? And it will take it and put it in packages. What do we call the packages? Transport vesicles. Transport vesicles transported to what? To the finishing deck or the finishing unit. And this unit is going to put it in a nice package, going to do like finishing, right? If you get a TV, for example, you don't just take it from the machines and export it out, right? No, you have to take it, put colors on it, finish it, put it in a nice package, and then export it, right? This is the Golgi apparatus. So the Golgi apparatus is going to modify, renew, package. Imagine yourself preparing something for export. What do you think you will do? Right? Renewing, you're going to finish anything that's not supposed to be there, you will take it off, right? Put it in a nice package. So this is basically what the Golgi apparatus or Golgi complex is doing. It takes these transport vesicles from the endoplasmic reticulum and package it, modify and package, and finish. Look at this picture. From this side, do you remember what is this? What's the transport vesicles for? Coming from where? From the rough endoplasm? Yes, rough ER. So they're going to transport it and send it to what? To the Golgi apparatus. As it is going in the Golgi apparatus, look what's happening here. It's going to make it in another product that's called secretory vesicles. Do you see the difference? Transport vesicles transporting the proteins from rough endoplasmic reticulum to the Golgi apparatus. What's the final product ready for export? Secretory vesicles. It contains proteins, but ready for export, ready for to be secreted out, which is exporting. This end here is the entrance, and this is the outlet. So transport vesicles and secretory vesicles. So look at this. This is the whole entire process. DNA contain the uh, manuscript, contain the instructions to make proteins. Okay? I want protein number one. Okay, you'll make a copy called messenger RNA. This is a copy. What's a messenger RNA? A copy for a protein. You do not, the DNA does not leave the nucleus. Is that clear? The DNA is very important and very expensive. You cannot lose it. You cannot send it out of the nucleus. You as a manager just stay in office. DNA stay inside the nucleus. It never leaves. Clear? So how can we make how can we send the instructions? I want to make protein number one. Okay, get a copy of the instructions of protein number one and send it to the machines, to the factory. Clear? What is this instructions? This part of the instructions. Messenger RNA. Okay? So I want to make protein number one. Here is the DNA, okay? One, two, three, four, five, six, all different types of proteins. What do you want? I want to make protein number one. Okay, protein number one is this instructions here. I'm going to take a copy of this, and I call this copy messenger RNA because it will carry the message. What's the message? Instructions to make the protein. So it will take it out. This is small enough and not the most important thing. It's just a copy, and it will leave the nucleus. There are pores. Leave the nucleus and go to the ribosomes to make the proteins. Clear? Can the DNA leave? No, it cannot. Two reasons. Number one, it's too important to leave. You don't send like your original script outside of the storage and or outside of the office and send it everywhere, right? You don't do that. You keep it inside, but you can take a copy of what you need. Clear? What else? How do you prevent DNA from going out? The nuclear pores, the pores, remember the nucleus is surrounded by a membrane, okay? The pores are small enough for the messenger RNA to leave because messenger RNA is tiny, but DNA is too big. This is like if you have a truck inside this room. Can you take it out of this door? No, but one of us can go out. One of us is messenger RNA. The truck is the DNA. It can't leave. Too big to leave. So you got the message out. You go to the ribosomes. Ribosomes start to make the proteins. And the protein, if it is rough RNA, it will go inside the rough endoplasmic reticulum until it goes into the transport vesicles right here. And look at this. This is the transport vesicles. It goes into the Golgi apparatus. And this surface, your entrance, is called the forming phase. You enter inside, and when we do it a well package, it's packaged, it looks good, everything is okay. It goes from the other side. It's going to leave from the other side in the form of secretory vesicles. Secretory vesicles. Is that the only function? We need to finish as much as possible, okay? Still have time. So is that the only function? It's just to get the proteins from the rough endoplasmic reticulum. You package it and you put it in a nice package and you call that secretory proteins. No, this is not the only function. There is another function. What is it? You make another organelle. Do you guys remember I told you organelles? Some of them are membranous and some of them are non-membranous. We talked about the non-membranous and we're talking about the membranous now, right? We talked about ribosomes, endoplasmic reticulum, and Golgi apparatus, didn't we? The next one that we'll talk about is called lysosomes, and here is where we make it. So this is the other function. It's not only packaging. You make lysosomes, but listen, these lysosomes are not ready to work yet. We call it inactive ribosomes, primary ribosomes. I'm sorry, not ribosomes. I mean lysosomes. Inactive. It doesn't work yet. Okay? We will talk about the lysosomes, but you have to know this. Like Golgi apparatus, both of them. Packaging to make secretory vesicles for export of the protein. This is one. The other one is to make the lysosomes inactive yet. It's not active. It's not ready to work. Okay, so this is step by step what I mentioned already. Go messenger RNA, which is a copy, it leaves. Look at the pores, how small it is. Compare, look at this. Compare the DNA to the pore. Huge, right? You cannot get it out. But look at how small the messenger RNA is. It's too small and it can easily leave. Okay, so it leave, go to the ribosomes, whether it's free or on the surface of the endoplasmic reticulum, and you are going to make proteins inside these ribosomes. And this is the protein, by the way. You know what? Every one of those, amino acid, amino acid, amino acid, you see these spherical structures? It's a polypeptide. Then it go to the rough endoplasmic reticulum in order to package it, and it leaves from the other end, which is the exporting end, in the form of secretory vesicles. Here is the forming side, and this is the maturing side. You enter from the forming, which is your entrance, right? You enter from the entrance. What's the entrance? The Golgi apparatus. The forming phase, okay? The forming phase. You go inside. You do the job, packaging and everything, and then you leave. What do you call the exit? Maturing, because it's mature. What's leaving here is mature. What do you mean mature? Ready for export. Okay, so this is the other phase. You also make lysosomes, as you see here. Here are the secretory vesicles. Look at this. This is something also that you need to know. These secretory vesicles, listen, secretory vesicles are not going to leave the membrane and continue outside like this. No, it's going to unload and go back. Like if you want, um, like some rocks, okay? I don't take my truck and give you the rocks and I keep the truck with you, right? I get the truck into your house. Where do you want the rocks? Right here. And then I will unload it and go back, right? So these vesicles, when they go to the membrane, they fuse with the membrane, unloading, and then it's going to reform the membrane. Here are the lysosomes. Lysosomes is lyso, this is part, and then somes. Lysome coming from lysis. Does anybody know what's lysis? Lysis, breakdown. Lysis means breakdown. Lysosomes, the bodies that break down. And these lysosomes, how do they break down? This is your recycle pen, okay? This is your shredder. Lysosomes is your shredder. So if you wanted to break down something, you send it to the lysosomes. The lysosomes are going to fuse with it, and they be activated this way, and they're going to break it down using their enzymes. It's full of enzymes, and that's why it's membranous, by the way. It's membranous. You know why lysosomes are membranous? It contains very powerful enzymes. You can use it, send something to it to break it down. But guess what? Guess what? If these lysosomes are not membranous, the contents are released, it is going to destroy the whole cell. And this actually happened, by the way. When the cells reach their lifespan end, they live for 3 months. After 3 months, lysosomes open. They are going to destroy the cells. Okay, but these lysosomes, when you make it in the Golgi apparatus, we call it primary lysosomes, which is inactive. When are the lysosomes activated? When it actually interacts with something that you wanted to break down. Okay? So what exactly do you break down? Anything that we don't need. Anything in the cells that we don't need, right? What do you put in the garbage? It's exactly like this. What do you put in the garbage? Anything, right? Papers, dust, plastic container, anything, right? Anything that you don't need, you just get rid of it. Put it in the garbage, right? This is your garbage, lysosomes. This is the garbage container. So it's going to break it down. And here is the whole process. You make the primary ribosome, lysosomes that's not ready yet. Look at this one here. This is mitochondria, let's say, mitochondria. The mitochondria is damaged. It doesn't work anymore. How can you get rid of it? Send it to your garbage disposal, fuse, become active. We call it the secondary, and it will break it down and get rid of it. And this word is what I just mentioned, autolysis. What's autolysis? Autolysis. I mentioned lysis. What's lysis? Breakdown. What's auto? Automatic or self, right? So you break down yourself. Does that really happen? Yes, this is what I just mentioned. Lysosomes are in a membrane. Only if you get something to the lysosomes, it will be broken down. But if you open the lysosomes? Like the enzymes are going to be like all over the cells, destroying the cells. Do we actually need to destroy the cells? Yes. It's called autolysis. This is programmed cell death. It's programmed. Red blood cells. It worked for 4 months. Can I put it aside after the 4 months? No. No, you have to break it down. It breaks down itself. It's going to send the signal, lysosomes. All lysosomes, all of you, open and throw your enzymes in the cells. That's going to break down the cells. Yes, it's over. Just get rid of it. This is called autolysis. Sometimes you call it apoptosis. Peroxisomes, the name peroxy. You have a question? Because when the lifespan is over, it's useless. That's number one. The cells at this point, like, don't you lose your skin cells on a periodic basis? Don't you lose the cells inside your stomach? Renew it. So if it is not working anymore, it's old, it's not working. Why do you want to keep it? So get rid of it, right? Because if it's not only that's useless, no, it can actually be harmful if you keep it. Peroxisomes are simply zomes. Somes means cells, bodies that contain peroxy, which is hydrogen peroxide. Hydrogen peroxide. Peroxisomes. Somes, bodies that contain hydrogen peroxide. Okay? Which work as a catalyst. Mitochondria is the next one. And the most important thing that I want you to know about mitochondria is powerhouse or energy. Powerhouse or energy. Good enough. Powerhouse or energy. This is where we make our energy in the mitochondria. The inside is called matrix, and there is a membrane outside folded in the form of crystal. Okay, but what's the most important? Making energy. Powerhouse in the form of what? ATP. Clear? That's what I want you to remember. Mitochondria, powerhouse, you make energy in the form of ATP. Okay? All right. Can you guys continue more? Or can we continue a couple more slides? Okay. So how exactly do we produce energy inside the mitochondria? There is something called glycolysis. I get the glucose and I break it down and I get energy out of it. Glycolysis. What's lysis? Third time. What's glyco? Sugar. You break down the sugar and you get energy out of it. Glycolysis. So you get the glucose inside and you break it down. Break it down into what? Into something smaller called pyruvic acid and get energy out of it. And then the pyruvic acid, the remnants, will go to the mitochondria and you're going to make another cycle that's called citric acid or Krebs cycle or TCA, tricarboxylic acid cycle. And this is going to break down the remnants more. Is that it? No, there is a third round called electron transport chain also inside the mitochondria. Let me say it again. This is your cells, okay? You need glucose to go into the cells. You need to break it down, get energy out of it. You do it in 3 steps. First step, get the glucose inside the cells. Now it's in the cytoplasm. Clear? Break it down into pyruvic acid. Get energy out of it. How about the pyruvic acid? It still contains a lot of energy. You don't throw it away. You send it to the mitochondria and you do 2 sets of reactions. to get every single piece of energy out of it. It's not enough to just get the glucose and cut it into two pieces. Every piece contains a lot of energy, right? So you split it in the cytoplasm, yes, and you get some energy, a little bit. Now I have pyruvic acid. Pyruvic acid contains a lot of energy. You don't throw it away. Send it to the mitochondria, and the mitochondria know how to get every single piece of energy out of it in two steps. The first one is called citric acid cycle. You get some ATP out of it. And the last one is electron transport chain. You get the electron and keep transporting it until you get the last energy out of it. So what determine which of the three are you going to do? What happens to the glucose when it enters the cells? You go to the cytoplasm. What do you call it? What are you going to do in the cytoplasm? Glycolysis, right? Glycolysis. This step does not need oxygen. Glycolysis. Are we following? And you end up having pyruvic acid, two sets, two pieces, pyruvic acid. Pyruvic acid still contain a lot of energy, right? You're going to send it to mitochondria. What's the problem with the mitochondria? Mitochondria doesn't work without oxygen. You have to give it oxygen to work. What if I don't have enough oxygen? It's not going to work. And you only do the first step. That's why we call it aerobic versus anaerobic. Aerobic, air, if you have oxygen. So the mitochondria will work, and it will give you the Krebs cycle and electron transport chain. How about the very first part, like splitting glucose only in the cytoplasm? It doesn't need oxygen. We call it anaerobic. Did you get the difference? Aerobic, air, oxygen. So the first part is anaerobic and the second part is aerobic. Look at this. Glucose. The yellow here is the cytoplasm. Enter the glucose. Get energy, and what you end up having in product? Pyruvic acid. Did I need oxygen here? No, it's in the cytoplasm, no oxygen needed. Now you have pyruvic acid. You don't throw it away. If you have oxygen, send it to the mitochondria. And the mitochondria will pass it into two steps, citric acid cycle, and then electron transport chain to get the rest of the energy out of it. So what happened in the mitochondria is called aerobic, which is the two steps. What happened in the cytoplasm? Glycolysis is anaerobic. Membrane flow, this will be the last thing that I'm going to talk about. Membrane flow is what determine what cross the membrane goes in from outside to inside or from inside to outside. How can you cross the membrane? The membrane flow will depend on different things. Depending on the shape and energy and others. And the same membrane flow happen through the membranes of the organelles, and we will discuss it later. Nucleus is the largest organelle. It's also membranous. It contains a double membrane, just like the cells. Do you remember the cell membrane? It's two layers of phospholipids, right? Same thing in the nucleus. And what's the importance of the nucleus? DNA. This is the manager's office, DNA. DNA is a manager, right? So it contains the DNA. And it's a spherical structure, but is it like completely closed? The DNA does not communicate. No, there are pores. Remember the pores, the messenger RNA can pass through it. So here it is. Here is the nucleus. And look at the pores, these tiny pores here. And inside the nucleus, there is another one that's called nucleolus. Do not confuse those two. Nucleus, nucleolus. There is an O here that's different. Nucleolus is kind of nucleus of the nucleus. It's a nucleus inside the nucleus, and it has another function. What can go through these pores? Messenger RNA. Can the DNA go through it? No, it cannot. So there is the plasm of the nucleus is called nuclear matrix. There is a nucleolus inside the nucleus. And these ones, is it the same as the nucleus since it's inside of it? No. It's the center, but it does another function. What is that function? It makes RNA, ribosomal RNA. This is where we make it. And last word, nucleosomes. Nucleosomes means the bodies that contain the DNA. Basically, nucleosomes are, you got this protein called histone. You know that this DNA, it's not scattered. It's wrapped around histones, and we call that nucleosomes. It can be DNA wrapped around histone. It can be in the form of chromatin or chromosomes. Both of them, yes. DNA around histones. Is it chromatin or chromosomes? It depends. If it is the cells are not dividing, it will be loosely around it. Like if I give you something, I told you wrap it around it. You can just loosely like this, right? Because you're not dividing. You don't need to tight it. If you do the same thing exactly, but make it tight, we call that chromosomes during cell division, and we're going to stop here.